Alright everybody, welcome back to lecture 11 of PLPA 303. So this is the very last lecture. Woo! Finally made it, huh? Easy day today for the most part, just a lot of waiting. But we're going to go over a very important technique in molecular biology today. So first though, we're going to talk about systemic versus local infections. In a viral infection, a plant virus can either infect one part of the plant or it can spread to the entirety of the plant. When you get that spread, we call that movement systemic, or systemic infection, systemic spread. And here in this picture you can see this tobacco plant. The entire thing is covered in lesions. So we would say that this plant is systemically infected. Whereas local lesions are really only seen when a virus is contained, because the virus, a plant virus, is always going to try to spread to other cells, as it replicates. But if a plant is able to produce that hypersensitive response that we've been talking about the past few weeks, then the plant can kill those infected cells before the virus can spread, and therefore contain that infection and prevent it from being further infected. So you could say that's just a local infection or a local lesion. And you can see all these tiny little dots, similar to some of the tobacco plants we have in the back there. All these tiny dots were cells that were at one point infected, and the plant was able to kill those cells before the virus could replicate and spread. Alright, so now we're going to switch gears and move over to serology. So who knows what serology is? I'm sure you guys have heard it in like TV shows and stuff. Serology is just working with proteins, especially antibodies and other antigens. So serology is just a diagnostic tool for using antibodies, or using antibodies, I should say. Isaac, why don't you go ahead and sit over here a little bit. Let's see, where was I? So what's cool about antibodies is that you can use it for any type of organism, and I'll show you guys why that is in just a second. But it's all based on using an antiserum, which is just any liquid that contains antibodies to react with a specific antigen. An antigen is what the antibody recognizes, what it binds to. So more specifically, it's the shape of a protein. So each protein has its own specific shape depending on how it folds and which amino acids it's composed of. And this can determine what it looks like in 3D space. So in your textbooks, you always see proteins usually as something like a circle or something very simple. But in reality, they're very complex. They have a lot of folds, a lot of grooves, and every single one of them is very unique. And those unique spots that can be recognized by antibodies are referred to as antigens. So the antibody is produced in response to antigens, only in animals though. And these antibodies will recognize specific antigens. So that's the concept behind what we're going to be doing today, which is called ELISA. But first let's look better at the antibodies. So they're highly specific in what they can detect. If you guys have taken biochemistry, you'll know that you got a light chain and a heavy chain. You guys don't need to know what that is for this class. All you need to know is that they're kind of Y-shaped, and the prongs are the site which binds to the antigens. These are the sites that recognize the antigens. The reason they can recognize it is because each antibody, as I mentioned, is specific. So it's going to be kind of like fitting together a puzzle piece. You can't really fit two pieces of a puzzle unless they're meant to fit together. Well, it's the same thing with antibodies and antigens. For the most part, there is a little, there can be a little bit of overlap in that. But for the most part, antibodies are very specific, and the antigen binding sites of antibodies will recognize specific proteins. So how are antibodies produced? It's a very complex process, and I'll be honest, I don't even know completely how they're made. But for this class, all you need to know is the basic concepts, and that first we have to isolate some sort of antigen. In this case, we want to isolate TMB because that's what we're working with, right? And once we have that isolated antigen, we inject it into some mammalian animal. So we can work with rats, rabbits, sometimes even horses, I've heard. Chicks, that's what we're doing with COVID right now. We're injecting a whole bunch of little baby chicks with COVID, and then we isolate these antibodies that are produced because the mammalian bodies can detect these foreign antigens and can produce antibodies that recognize that specific foreign antigen. And that's how you get an immune response. I'll tell you guys later how plants do that. Plants don't produce antibodies, by the way. But we do use antibodies for plant-related research. So, okay, you inject TMV, in this case, you inject TMV into a rabbit or whatever, then you have to sacrifice it, unfortunately, and you isolate the antibodies that can specifically bind to the antigens of what we're interested in, which is TMV. And then what you do the serological testing, which is ELISA, enzyme-linked immunosorbent assay. So the way it works is you're each going to have a little tray with wells. So this here represents one well. And on that well you have antibodies stuck to the bottom of it. So try not to get anything into the wells. Normally we keep them covered up, but for this class it's fine. But don't go stabbing the bottom of the well with pipette tips or your finger or anything. Because the strips that you guys have, they're called immunostrips. Anything that says immuno in it usually has something to do with antibodies. So we've got these immunostrips with antibodies that can specifically detect TMV, which here we have represented as just this diamond-looking thing. And when we add some serum or anything to these wells that contains TMV, the TMV is going to be bound by the antibodies just because they're like the puzzle piece. They're going to stick together and they're going to stay stuck together. Once that happens, we then add a conjugate, which is another antibody that binds to a different part of the TMV, or a different antigen, I should say, but it's still part of TMV. So while we have TMV here represented as a diamond, in reality it's got more complex shape than that. So this conjugate's going to bind to a different part of the TMV, which is held to the well by this other antibody here on the floor. And attached to this antibody are a couple of enzymes, which we have represented here as an E. So if you guys recall anything, or enzymes are proteins that can catalyze a reaction. So after we add the conjugate that has these enzymes, we then add a substrate. And this substrate is going to interact with this enzyme. And when you get that interaction, a reaction occurs because the enzyme is able to catalyze a reaction. That reaction is going to change the color of the solution, and that's how you'll be able to differentiate between a positive or negative sample. Let me show you what I mean. So the first things first, we're going to have these empty immunostrip wells, right? They already have a blocking agent, so we're up to this step now with what you guys have in front of you. So all you have is just an empty well with an antibody for the most part. We're then going to extract some sap from your plants that you inoculated, and we're going to add it into the well. So here we have the green sap representing, the green represents the sap, and the TMV particle is again this diamond shape. So if there's any TMV floating around in solution, it's going to be bound to this antibody. Afterwards, we're going to wash everything away, and only the antibody should stick around because again, the antibody, only the TMV particle should be sticking around because it's bound to the antibody. So then we add the conjugate, which is going to bind to a different part of the virus, and you can see here that the top part of the TMV is different than the bottom part, so that's just a little better representation. Then we add the substrate, and if the substrate interacts with the enzyme that's stuck to the conjugate, then you get a color change, and that's how you know you have a positive sample. And on the bottom here, we have an example of what a negative reaction would look like. So we would add a sap, we would add the sap from a plant that is not TMV infected, and you can see here there's no TMB particles. So when we wash this away and we add the conjugate is floating around in solution because it can only be stuck to TMB particles, whereas this antibody down here is stuck to the well. So when we wash away the conjugate and then we add the substrate's not going to change color because there's no enzymes around to interact with it. So it's all dependent on this conjugate sticking or binding to the TMB particle so that you can get that reaction. And that's how you get this difference here. Up here you get a change in the color of the substrate, down here you don't. And this type of ELISA is called sandwich ELISA. There's different types of ELISA. All ELISA is just an antibody stuck to a well, and then you can start adding different stuff up here. But for this purpose, this is called a sandwich ELISA. And that's what we're going to be doing today. Are there any questions on that? Nope? All right. So today you got two assignments. The first is to take observations on your plants that have been infected for 2 weeks now with TMB. And then the second part is to do the actual ELISA. So there's going to be a lot of incubation steps, a lot of waiting. The immunostrips we're going to put them in a certain order. The wells are all the same, but we need to have a positive control and a negative control, as you saw in your previous experiment, so we have something to compare it to. And you guys should have 3 samples, so we're only going to be using 5 of the wells today for the immunostrips. The positive sample we have in front of your desk, which is just TMB that has been isolated. Your negative sample is just going to be water. And then your 3 samples are going to be your tomato plant and then your 2 varieties of tobacco plants. And those will be your 5 samples. Well, make sure you write down either on the wells or write down on a separate sheet of paper what's in each well. It doesn't matter which order you load them in, just make sure they're all together. And next week we have our final exam. Make sure you have all your PPE that you bring with you. If you have any late assignments that you have turned in or you turned in after the due period, then the maximum you can get is up to 50% of what it's worth. And then we'll do some review here in class while we wait for some of the assignments. So during the practical, for the students that are here in person, for you online students, it's going to be a little different. I'll get into that after this. For the students that are here in person, we're going to label each one of the tables with a certain number, and each one will have a question. There's a map online of how the room is going to be laid out, and you guys are just going to be walking around. We'll either time you guys, like every minute or so, everyone moves one spot over. 64 questions and 24 stations. All your smartphones and your cell phones or whatever and your smartwatches will have to be put up here up front. And yeah, just don't cheat, please. Any questions on the exam? For the online students, you guys are just going to get another standard exam. So sorry about that, I find it a little boring. Any questions in here? No? Okay, let's get into the practical part.